basically these different geological layers are assemblages of different ecosystems. And the Cambrian ecosystem is a marine ecosystem. You're not going to find a rabbit, but I can't produce a Precambrian rabbit out of my hat and disprove evolution, but there are many questions which are raised by the fossils. And we don't have a rabbit, but we do have a fish called Metaphysis attenborough. Why they make these names so complicated to say. That's basically motherfish, named after David Attenborough. Now, motherfish is an interesting term. Why is it called a motherfish? Well, we'll come to that. Most fish, as you will know, lay eggs. But some fish give birth to live young. These are the sharks and the rays, which more often than not give birth to live young. And this is considered to be an advanced feature. So from evolutionary terms, you start off with a fish that lays eggs, and you evolve into a fish that lays live young, because you have to have a whole complicated system to keep your fish alive. And this is information coming from Nature, published May this year, no less, so it's hot off the press. But this fish, a Metaphysis, has a uterus, a yolk sac connected to the embryo and umbilical cord. And this was found in the fossil of this fish, which was dug up from the Devonian period. Right, so here we have a diagram, a picture of the fish actually giving birth. It's a modern fish, essentially a modern fish in the Devonian. And it's almost as good as a rabbit in the Precambrian. Not quite as good, but this shouldn't happen. And if you read the article in Nature, the authors are quite stunned that this advanced feature is present right at the beginning of the fossil record. And that's not the only example where the evolutionary story doesn't seem to tie up, because what you're finding here is very advanced features appearing right at the beginning of the fossil record of any particular type of animal. And birds are another example. The early bird is actually Archaeopteryx. Archaeopteryx is a bird. It's a funny bird. It's an odd bird, but it is a bird. And it appears in the Jurassic rock. So here we have our geological column, Jurassic period here. Remember that as we go to the next slide, because there is a problem with the story, because actually there's an even earlier bird than Archaeopteryx. And I haven't got a fossil of it because there aren't any fossils of it, but what we do have, and this was published in Nature in 2002, are footprints. Now, if you look at that, I think if you're unbiased, you're going to say they look like bird footprints. And they do look like bird footprints. And the authors of the paper actually admit these are Triassic bird-like footprints, actually in the era before the Jurassic. So they shouldn't be there, because, we know they shouldn't be there, because there were no birds in the Triassic. Okay. It's got 4 toes. It's got a backward-facing toe. So what we essentially have here is what appears to be perching birds in the Triassic, millions of years before Archaeopteryx. So what I'm presenting to you here is not, again, it's pretty much like the rabbit. It shouldn't be there, because birds hadn't evolved at this time. So what you find as more and more fossils are dug up of any particular type of fossil, any particular species or group of species, is that you tend to find that as we get more and more evidence, we find that all the advanced features which are present today were also present right at the beginning of each species. And that creates a bit of a conundrum for the evolutionists, because they have sudden evolution of all these advanced features, which isn't very Darwinian. So there are problems in the story, the evolutionary story. There are gaps, there are problems. So let's move on. Dawkins then addresses irreducible complexity, and we need to address it. He makes a rather bold statement and says, no attempt is made to demonstrate irreducible complexity. Well, I've got a book in my briefcase here called Darwin's Black Box, where a gentleman called Behe spends quite a number of pages demonstrating irreducible complexity, so that's not true to start with. Behe does demonstrate irreducible complexity, and he uses the flagella motor of the bacteria as an example. So I want to look at the evidence. I want to follow Professor Dawkins' advice. Let's look at the evidence for the irreducible complexity of the bacterial flagellum. But before we do, I just want to remind you that not all complex objects are irreducible. There's a difference. We need to be careful here. The eye, for example, is very complex, but the eye is not irreducible, because you can take bits of the eye away. You can take out your lens inside your eye and you can still see. So your eye is not irreducibly complex. You could take out various bits of your eye and still see things. Not so well, but you can still see. So it's not irreducibly complex. And that's an important point. And we don't claim it is. No creationist I know, no creation biologist claims that the eye is irreducibly complex. They claim it's complex. They claim it can't evolve. Different story. But most of us do claim that bacterial flagella are complex and irreducible. You can't take bits away, one bit away, and still have a functioning bacterial flagella. But we need to bear in mind, we need to separate complexity from irreducibility. A very simple system can be irreducible, and we'll come back to that. Well, let's just look at the bacterial flagella, in case you're wondering what I'm on about. It's basically a whip-like organ that bacteria have, quite a number of bacteria have, that allows it to move through liquid media. And here we have a picture on the top here of a salmonella bacteria that causes various different intestinal infections. And it has flagella which allow it to swim. And below you have a diagram of the flagella motor, which is the motor that drives the flagella, makes it rotate, and makes the flagella operate like a propeller. And it's got all the components of a motor. It's actually a very complex rotary motor, has about 40 different components. Now, this is where we have to be careful about complexity and irreducible complexity. You can take most of those components away. If you look at all the different types of flagella motor that there are, you end up with about 20 components that are common to all, so we might suggest that these 20 components are the minimum you need to have a working bacterial flagella. So here is an example where you can't take away any more parts. If you take away any more parts, it stops functioning. So it appears to be irreducibly complex. But the evolutionists aren't going to lie down that easily. They say, well, we can evolve a bacterial flagella, we can evolve the motor. We can just have a process where we can go back to the good old Darwinian principles, slow gradual increase in complexity starting from some basic components. And we'll be able to keep it functionally useful at every stage because it has to be functionally useful, it has to do something at every stage. Otherwise, natural selection can't operate. And by gradually changing the function of some components, we can evolve to a more complex bacterial flagellum that we see today. And this process is basically called co-option. You co-opt existing proteins that are in the bacterial cell, and you change them around a bit and make them into slightly different proteins which do slightly different functions. So this is what the evolutionists would say happens, and they appeal to something called the type 3 secretory system, which exists in bacteria, which is simpler than a bacterial flagellum, and they claim that you can evolve from a type 3 secretory system to a flagella motor. But the question is, which comes first? And even evolutionists have a problem with the story that the type 3 secretory system came first, and they suggest that the evidence supports the counter-argument that, in fact, the type 3 secretory system is a degenerate form of the bacterial flagella motor. But the evolutionary explanations start with this type 3 secretory system. But we still have to ask the question where it came from, because it's still quite complicated. So where did it come from? Well, they say we take the ATP synthase and a simple protein export system, put them together, and we end up with a type 3 secretory system. Well, I'm not going to go into detail about what all that is on about, but the ATP synthase is not a simple machine. It's quite a complicated machine, and a simple protein export system also is not a simple machine. It's composed of many different proteins. So both these parts are also complex. So where do they come from? So the evolutionist still has a lot of explaining to do. But I want to look a bit more depth at the evidence, because they claim that maybe we haven't got all the evidence we need, but if you look at the proteins that you have in the bacterial flagella motor, and you look at the proteins available in different other systems, they appear to be similar. They appear to be homologous. They appeal to homology. They say we have similar proteins doing slightly different things, modified slightly so that they can do a different job. So this is the source of the protein components of the bacterial flagella. So if we, for the sake of argument, allow them their type 3 secretory system, which evolves into a bacterial flagella, we'd expect to find homologous proteins, which are variations of existing proteins. And it is claimed in the literature that all but one of the essential proteins is a homologue of another protein. So now I need to really explain what I'm talking about, because unless you're used to these terms, you probably haven't got a clue what I'm saying here. Let's go back into cytochrome C. Cytochrome C is a protein which exists in just about every creature on Earth, because it's involved in a very important metabolic process. So you can go to just about any animal cell and extract cytochrome C, and you can look at its amino acids. Proteins are made up of amino acids. And you can compare the cytochrome C from different species, and you can actually look at the differences and say, well, cytochrome C from a human being and an ape is very similar. Cytochrome C from a human being and a horse is less similar. Cytochrome C from a bacteria and a human is very different. We can create some sort of evolutionary tree by looking at the differences. And that's what they do. And here we have a diagram of cytochrome C. And here you have the different amino acids represented